In 1958, Meselson and Stahl were two scientists who conducted an experiment in order to determine the nature of DNA replication. This was also known as the beautiful experiment. They had three initial hypotheses that they believed could have been viable explanations for DNA replication. The first is known as conservative replication, which is when two new copies are made of a DNA sample and one is the exact same, but the other is entirely new because of different base pairings. The second is dispersive replication, which is when the resulting two copies of DNA are different from the initial one due to mixing parts of the original strands along with new aspects. Finally, there is semiconservative replication, which is when one of the two copies has a strand that is completely new and the other strand is a mix of the original along with the new one. Meselson and Stahl's experiments involved growing E. coli in a 15-nitrogen nutrient broth for many generations and then sampling the DNA. After that, they would transfer these collected DNA cells to a 14-nitrogen nutrient broth and then let it replicate for 3 more generations with sampling in between each one. The DNA samples were then measured using density gradient centrifugation, which is when the different components of a sample are separated based on their density. They found that after the first sample, the DNA was at an intermediate point, but would then separate into 2 distinct areas in the nutrient broth. If conservative replication was the correct explanation, then there wouldn't have been an intermediate mass of DNA, so Meselson and Stahl were able to eliminate that hypothesis first. Dispersive replication was a bit trickier to get rid of, though, since in the first generation, both dispersive and semiconservative showed the DNA having an intermediate density. However, from that point on, the dispersive replication would have still only had one mass to measure within the nutrient broth. That is not what happened, though, and the DNA equally separated into 2 groups with different densities. This led to Meselson and Stahl to conclude that DNA replication is semi-conservative, since it showed signs of having entirely new strands along with mixing both the old and the new.